So our next speaker is Bridget Barber, and Bridget's from Queensland, an ID physician. Bridget's going to talk about the now unfunded National Clinical Evidence Taskforce. And if anyone feels the incentive to write an email, please do it, because as Bridget will highlight, it's a very important body. Thank you.
Thank you, Lou. Thanks everyone for having me. So I'm going to talk about the drug treatment guidelines of the National Clinical Evidence Taskforce. There is quite a bit of overlap with James's talk and with the discussion that we've just had. So by the end of my talk, everyone hopefully will have a very good understanding of the drug treatments of COVID-19. So I'm just going to start with an overview of the task force and go over how the guidelines are developed, discuss some of the limitations and challenges, give an overview of the current treatment recommendations and obviously a bit of overlap with what James has been talking about, and then just at the end show you some of the other clinical tools that the task force has developed. So the task force is a multidisciplinary collaboration of 34 peak professional bodies across Australia whose members provide clinical care to people with COVID-19, and the member groups are listed on the right of the slide there. And the role of the task force during the pandemic has been to undertake continuous evidence surveillance to identify and rapidly synthesise emerging research, to provide national continually updated evidence-based guidelines for the clinical care of people with COVID-19, and to offer a unified national clinical voice providing guidance to Australian clinicians. So this is an overview of how the recommendations are developed. So the task force has a clinical evidence team who search through the many thousands of studies to identify the relevant high quality research. And in the case of drug treatments, this is restricted to randomised controlled trials, as James mentioned. And the evidence team evaluates the studies using the grade methodology, which I'll go through in a subsequent slide, and prepares a summary of the evidence, which is then presented to the relevant panel, and the one that I co-chair is the drug treatment panel. The panels then review and discuss the studies and come up with a treatment recommendation, which then goes to the guideline leadership group and the steering committee for approval, and is then published on the website. And to date, there's been over 24,000 studies screened, over 200 recommendations developed, and the guidelines have been very widely used. And as Lou mentioned, unfortunately, the funding came to an end at the end of last year, so it's uncertain. The future of the task force is uncertain at the moment. We do have some funding to continue at least until the middle of this year. So I just thought I'd go through the types of recommendations that we can come up with. So we can come up with a strong recommendation, and these are colour-coded, and a strong recommendation generally correlates with a high certainty of evidence that there's a benefit from the intervention. It's much more common for us to come up with a conditional recommendation for a drug where there's moderate certainty of evidence that there's benefit from the intervention. We also very commonly advise to only use an agent in the context of clinical trials, and we give this recommendation where there's low certainty of evidence with insufficient evidence to determine if the intervention is effective or not. We can give a recommendation to not use an intervention where there's high certainty of evidence that there's no benefit from the intervention. Less commonly, we give a conditional recommendation against using an agent, and we don't use this one so commonly. Normally, interventions, so for example, ivermectin or hydroxychloroquine, sat in the only in research category for until we had sufficient evidence accumulate, and then they moved to the not recommended box. So in fact, molnupiravir is the only agent that has received this conditional recommendation against. And we can also give a consensus recommendation where we don't really have sufficient evidence to give one of the other recommendations, and this is really an opinion-based recommendation. So the evidence is assessed according to the grade methodology, which many of you will be familiar with. It provides a transparent and systematic approach for evaluating and presenting the evidence. For each intervention, all the clinically relevant outcomes are separately evaluated. So for example, the outcome of reducing hospitalisations will be evaluated separately to the outcome, for example, of improved time to recovery. And the certainty of evidence is assigned for each separate outcome. Where relevant evidence is considered separately for different patient groups, so for example, considered separately for those with severe COVID or those with mild COVID. According to for the drug treatments, only randomised controlled trials are considered, and the data from the new randomised controlled trials are combined with data from previous randomised controlled trials, so essentially it's a living meta-analysis. According to the grade methodology for RCTs, the certainty of evidence begins as high, so the default is a high certainty of evidence, and then it's downgraded for risk of bias, imprecision or inconsistency. The recommendations are based primarily on the strength of the evidence, and so factors not incorporated include factors such as cost, availability and accessibility. So this is an overview of all the therapeutics for which there has been a recommendation developed. So the ones in the box in the middle there are the recommended drug treatments, the corticosteroids, the antivirals, now only 2 that are recommended by the Taskforce, the monoclonal antibodies, there's 4 listed there, but obviously very significant caveats in terms of the emerging variants, and the immunomodulators, as James has already mentioned, there's actually now 5 that have recommendations. Sarilumab isn't available in Australia, but it's still listed there, and of course tocilizumab and baricitinib are the 2 that we're most familiar with. Then there's the list down the bottom there of the treatments where we had sufficient evidence to say that they don't work, such as hydroxychloroquine and ivermectin, and molnupiravir is on the bottom of the list there. And as I mentioned, that's a conditional recommendation against its use. And then the very long list on the right there where we have insufficient evidence of whether they work or not, so that they're just only in research recommendation. So this is just an example of the evidence summary that accompanies each recommendation for the Taskforce. So this is corticosteroids versus standard of care. The top row there is the outcome of all-cause mortality in adults requiring oxygen. The second column there is the relative risk, and it provides the information on the number of studies and the number of participants that the analysis is based on. The middle column there gives you the absolute effect estimates. Then there's a description of the certainty of evidence and a plain language summary at the end. So corticosteroids probably decrease death at day 28 in adults who require oxygen. And then there's a similar analysis for separate outcomes and also for separate patient groups. So that bottom row there is all-cause mortality in adults not requiring oxygen. So this was actually the first recommendation that we came up with. And in the context of the time, with an unvaccinated population and reports of high mortality rates, we gave this a green, strong recommendation to use dexamethasone in adults with COVID-19 who require oxygen, and then a conditional recommendation against using it in adults who don't require oxygen. And then there's a whole lot of text that accompanies the recommendations that gives you all the information on the studies on which the recommendation is based. So moving on now to some of the challenges and limitations. So these are evidence-based guidelines, so they're limited by the quality, relevance and applicability of the randomised controlled trials. So the study populations very often don't match the current situation. Many of the studies were done during the time of the Delta variant. Many of the studies didn't include vaccinated patients, so particularly the studies that have evaluated treatments for mild COVID. Many of the studies have only a small number of immunocompromised patients. And then there's been the difficult challenge of how to deal with in vitro data when we're only really supposed to evaluate randomised controlled trials. So we have increasingly relied on consensus recommendations, which, as I mentioned, are opinion-based recommendations rather than strictly based on the evidence. And we have also tended to add increasingly lengthy remarks to the recommendations. So just to show you an example of that, this is the Paxlovid recommendation, where we have a conditional recommendation to consider using Paxlovid in unvaccinated adults, and then there's an asterisk there with a footnote that explains that the reason that the recommendation refers to unvaccinated adults is that the study that this was based on, which is the EPIC-HR study, excluded vaccinated patients from the study. So then we have a consensus recommendation that advises that Paxlovid can also be considered in at-risk vaccinated patients. And then there's also a statement there that the study was conducted before the Omicron variant. So there's little data regarding the effectiveness of Paxlovid specific to the Omicron variant. You've seen this a number of times now. James just showed this figure, and James wasn't here yesterday, but it was actually also shown yesterday, which is good because it means I don't have to talk through it. But obviously it's become increasingly complex in terms of the emerging variants. And in terms of the guidelines, this is particularly relevant for the treatment guidelines for the monoclonal antibodies. This is a great website that was just brought to my attention recently, where you can search for your antibody of interest and then search for your variant of interest, and it gives you all the in vitro studies that have evaluated the activity of the monoclonal antibody against whatever variant you're interested in. And, you know, I'm not a virologist, despite the comment yesterday that we'd all become virologists. I don't claim to be a virologist, but, you know, even I can see that the available monoclonal antibodies all look pretty rubbish against all the emerging Omicron variants, unfortunately. So previously we had dealt with this by, you know, leaving the initial conditional recommendation there for the monoclonal antibodies, but then just adding a statement in red that, you know, drew attention to the fact that, you know, they were likely to be ineffective against the emerging variants. You know, this I think really needs to be further reviewed at the moment in view of the recently emerging variants. There is an in vitro working group that advises the Taskforce in terms of how to interpret the in vitro data, but really at the moment I think, you know, as has been mentioned already, it doesn't look like that, you know, we have effective monoclonal antibodies at the moment. So additional challenges and limitations, we don't have very much data about the relative effectiveness of recommended agents. So there's generally no head-to-head studies comparing recommended agents. For example, no head-to-head study comparing remdesivir versus Paxlovid or tocilizumab versus baricitinib. One exception, which I'll just show you on the next slide, is that there has been a head-to-head study evaluating dexamethasone versus baricitinib. We don't have a lot of data on the use of agents in combination, and we don't have a lot of data on what to do in highly immunosuppressed patients. So this is just one study that I've put up because it is a rare example of a head-to-head study, which was a study done in the UK, done in hospitalised patients, just over 1,000 patients who required oxygen but not mechanically ventilated. They were all receiving remdesivir and they were randomised to either baricitinib or placebo or or dexamethasone and placebo. It was conducted during the time of the Delta variant. There was no difference in mortality, but there were significantly fewer adverse events in the baricitinib group compared to the dexamethasone group. So in terms of the Taskforce recommendations for the immunomodulators, you know, as we've mentioned, we've got recommendations for dexamethasone, tocilizumab, baricitinib, and now infliximab and abatacept. Our guidelines state that there's current evidence to suggest that dexamethasone is safe in combination with other immunomodulators, but that there's limited data about the safety of other combinations, with one exception being that the recovery trial did include patients who received dexamethasone plus tocilizumab plus baricitinib. However, due to concerns about increased risk of side effects with multiple immunomodulators, we do generally recommend that dexamethasone is used with one other immunomodulator. And then just recently we've added a statement that in view of this recent ACT4 study, that baricitinib may be considered as a standalone treatment in patients considered to be at high risk of side effects from dexamethasone. So this is my table of immunomodulators. James mentioned that he had reduced his talk to one slide per drug. I've reduced it to one table for all of the immunomodulators. The data you've mostly seen before, so there's a row there to list the number of studies and the number of patients that the analysis is based on. Obviously, baricitinib and tocilizumab, higher number of participants and higher number of studies, just the single study for abatacept and infliximab. So high certainty of evidence for baricitinib and tocilizumab for reduction of mortality at 28 days, and moderate certainty of evidence for abatacept and infliximab. We don't yet have data for abatacept and infliximab in mechanically ventilated patients, just because those patients weren't included in the active immunomodulator trial. And moving on to antivirals, this has been discussed already. That's just the table there that summarises the data that these recommendations have been based on. So the EPIC-HR study for the Paxlovid and the Pine Tree study for remdesivir, and molnupiravir is greyed out there because it's not recommended anymore. The certainty of evidence, so moderate for Paxlovid and remdesivir, that these agents reduce hospitalisations, and a high certainty of evidence for molnupiravir, that molnupiravir has no impact on mortality or hospitalisation. So just to go discuss a little bit more about molnupiravir, James has already discussed the Move Out study with the concerns regarding the differences in the interim analysis and the final analysis. And in fact, if you just look at the second half of the study, there was actually more hospitalisations in the molnupiravir group. So even based on this study, we only ever had a consensus recommendation for molnupiravir. Even before the panoramic study, we only ever recommended molnupiravir where the other agents, such as Paxlovid, weren't available. And then the panoramic study, James has already gone through the methods. Just a couple of things to point out. It was a relatively young cohort, so a median age of 56, 57. But if you look at the absolute numbers of those in the higher age groups, the numbers are still pretty big. So aged over 70 or 18 to 69 and extremely clinically vulnerable, that's still nearly 4,000 patients. So still a very significant patient number. Outcomes, as James has said, absolutely no difference at all in the primary outcome of hospitalisation or death. In the secondary outcomes, which was self-reported, time to recovery or alleviation of symptoms asked in quite a number of different ways. There was a significant benefit for molnupiravir in most of these outcomes. In terms of contact with health and social care services, a little bit more variable. Some of them favoured molnupiravir and some of them didn't. And this is the forest plot for the subgroup analysis. Just interesting to note, I think, possibly a trend here towards a greater benefit, an effect of molnupiravir in the highest age group, although not statistically significant, and in the risk quintile 4, it's actually right over the other side. It actually looked worse in immunocompromised, which I think you can't read too much into, and even if you just look at the subgroup who presented within 3 days of symptoms, there's still no statistically significant benefit of molnupiravir. And this is the time to first reported recovery. I think what's interesting here is, so the axes have been flipped around here, so this time on the right favours molnupiravir. I think what's interesting here is there's actually no heterogeneity at all within the subgroups. So if you look at the risk quintile, those at the lowest risk, who you perhaps wouldn't really expect to benefit significantly from molnupiravir, had the same magnitude of benefit as those in the highest risk group. So to me, I guess if there was a true effect of molnupiravir, I think I would be expecting to see some heterogeneity within those subgroups. So we've come up with a conditional recommendation against the routine use of molnupiravir for treatment of COVID-19 based on a high certainty of evidence that molnupiravir doesn't reduce hospitalisations or deaths. Molnupiravir may improve recovery, but this is a low certainty of evidence due to a serious risk of bias. So just in my last couple of minutes, I'll just show you the clinical flowcharts that the Taskforce has developed. So this is the flowchart for drug treatments for adults with COVID-19. So there's risk disease severity category along the top, and this is the Taskforce attempt to put all the guidelines, all the recommendations, into one spot. So you can just go to one spot and have all of the recommendations there. Evishield is still here, so this probably needs updating. And this is the decision tool for adults with mild COVID-19, and this is a tool intended to help clinicians work out whether their patients need any specific treatment for COVID-19 or whether they really don't need anything specific at all, based on whether they're immunocompromised or unvaccinated, and whether they're at particularly high risk due to advanced age or multiple risk factors, whether they're within 5 days of symptom onset, and then if you go down to the bottom of the chart, it tells you the recommended agents. And there are some notes over here which talk about some extra information, for example, monoclonal antibodies that have been removed because of emerging variants. And finally, this is a risk classification tool, which again is intended to help clinicians work out how at risk their patient is at progressing to severe disease based on whether they're immunocompromised, Aboriginal or Torres Strait Islander people, whether they have comorbidities, the time since their last vaccination dose, and their age along the top there. And in general, if you're in the red, highest risk of severe disease, then in general that would correlate to that the patient should be offered some kind of specific treatment. The green, they in general shouldn't, and anywhere in between in the orange groups, then the risk will depend on context, frailty and time since vaccination. And that's all, thank you. So just thanks to the COVID Taskforce and the rest of the drug treatment panel, and happy to take any questions.
Terrific. Questions, please. Rod?
Do you include any of the...
So thank you. I've got a couple of texts from people who actually appreciated the talk online. So I've been talking to people who are watching online and appreciate it. So thank you. But when it comes to long COVID, some of our cardiologists are using colchicine and other things when they're getting raised troponins, 3 weeks later. So do you consider you're advising treatment of acute COVID, or do you think any of your guidelines are about long COVID?
Our guidelines are all about acute COVID. As we've discussed earlier, we really don't have the data yet for long COVID. It's certainly part of the remit of the Taskforce to evaluate that evidence when there is evidence available. But at this stage, we don't have that data. Colchicine is on our not recommended list.
I saw that. And
yeah.
Can I just, I've got an online question that's just come through. Do you really care, they didn't say this, but from our point of view, do you really care whether someone's statin is more effective or not? How long do you got to leave a statin off or on Paxlovid? Because most of us just disregard that. But in theory, someone's statin is more effective for 5 days while they're on Paxlovid. Do we really care?
I think someone can come off their statin for a few days without significant risk.
Bridget, there's a group of people who get pneumonitis who then go on in weeks 3 or 4 to have persisting infiltrates that clinically are diagnosed as organising pneumonia, and we tend to use heaps of steroid in an unspecified way. Has the committee got any evidence or views about that?
I think it's a really good question, because I think it also, a similar question is in the severely immunocompromised, where we just see prolonged viremia, and often we end up giving a course of Paxlovid and then we give some remdesivir, and we really don't know what to do. I think it's a bit of an evidence-free zone. I hope going forward eventually we do have some more evidence to do it, to guide us what to do in those situations, but I think at the moment we don't.
Yep. There's a question. Alan.
Yeah, my question is about what do you do when you're not really sure? There's evidence of absence, and that's pretty clear. But there's a lot of situations where there's absence of evidence. So the case I'd make now is that the disease is different. The original trials were against severe pneumonitis and prevention of progression to that and dying from that. But we don't really see that anymore. But we do see people still being hospitalised with, geriatric syndromes and falls and delirium and so on. So in that situation where we're all, vaccinated, previously infected, is any of this? Do we have evidence to back any of this anymore? Does Paxlovid actually, do anything, because we don't get pneumonitis anymore? So what are we stopping prevention too?
I don't really have an answer. I think it's a really good question, and I tried to bring that up, when I discussed some of the challenges, because that's exactly right. The panoramic study is including Paxlovid, so I think we're all sort of eagerly awaiting that. Hopefully that will give us some information about whether Paxlovid is beneficial in vaccinated Omicron patients, or whether, like molnupiravir, is not. But all we can do is go on the evidence that we've got. I don't really have an answer in terms of what to, It's just an unavoidable limitation of these guidelines, and the situation is changing so quickly.
Bridget, Phil Britton, thanks very much for all your work on this. I know it's been a huge effort for everybody on these panels. I want to say, Alan Cheng, welcome to being a paediatrician. Because we've got no evidence, we've been making it up the whole time. But my question is about the consensus recommendations, which really do actually make up quite a lot of what comes out of the Clinical Evidence Taskforce. And what I don't understand is there a similarly robust methodology around generating consensus within the Clinical Evidence Taskforce? Or do you do what I do, sit around and think, oh, well, let's have a go?
I think it's an evolving issue that the Taskforce is continuing to work on. Mostly it's been we sit around and have a chat and work out what we all think, it's just an opinion based. For the emerging variants, as I mentioned, we've got the in vitro working group, which have been guiding us, and so that informed some of the consensus recommendations. No, it's not as robust a process. I'm not sure that it can be as robust a process. and we don't have a systematic process for evaluating observational data.
If you discuss a topic and there's a lack of consensus, do you publish that?
The task force is pretty firm on having consensus.
Right,
There's
right.
Some people in the audience would
Very,
know.
okay. So it's a very
So the
positive.
mono-peer review recommendation took us a long time
All right.
for that reason. It's the idea is that it's 100% consensus,
All
which can take a while to get to.
Good. All right, Bridget, wonderful. Thank you.